Now we're going to talk very quickly about G6PD deficiency. This is deficiency of glucose 6-phosphate dehydrogenase enzyme. This is part of the hemolytic anemias. Here, in fact, we have extravascular and intravascular hemolysis. So, due to the extravascular hemolysis, sometimes the patient can present with splenomegaly, hepatomegaly, bilirubin gallstones, ictericia, and for the intravascular hemolysis, would present with increase in the haptoglobin and decrease in the haptoglobin, increase in hemosiderin, and the hemosiderin is deposition of iron, ferritin in the tissue. Among those tissues could be the kidney. The tubular cells of the kidney, they pick up the hemosiderin and they can get like intoxicated, so they could be kidney damage. Actually, for G6PD deficiency, there is no treatment, just avoidance of the stressors. So the stressors would be sulfadrogs, nitrofurantoin, infections or conditions of stress like infections, sleep deprivation sometimes. Other medications include dapsone, rasburicase, usually in children, and quinine, an anti-malarial agent. Now, the pathogenesis is mutation in the gene that codifies for glucose 6-phosphate dehydrogenase enzyme. This enzyme is the one that prevents oxidative stress indirectly due to this is a enzyme as part of the exose monophosphate shunt that converts glucose to glucose 6-phosphate. This is by exokinase, and then glucose 6-phosphate is converted to phosphogluconate by the glucose 6-phosphate dehydrogenase enzyme. Now, here there is production of NADPH. This NADPH is used in the prevention of damaging the cells in a oxidative stress. That is, let's say that there is oxygenated water, H2O2, This is toxic for the cells, is always taking electrons from membrane of the cells or phospholipids. With this we kill the bacteria, but also is damaging the cells. So we have the molecule glutathione. Glutathione is in glycine, cysteine, and gamma-glutamine cysteine glycine. This is in the reduced form. It has a sulfhydryl groups, so S and H. When interact with the oxygenated water, two glutathione molecules and one oxygenated water will convert into H2O, and two glutathiones together, the sulfide groups will be joined, and this is called reduced glutathione. Here, oxidized glutathione, because it was oxidated due to the oxygenated water. We can revert this, so passing from oxidized to reduced glutathione. The enzyme that does this is glutathione peroxidase or catalase, and the one that returns this is the glutathione reductase. And here we need NADPH. This enzyme requires NADPH, and this NADPH is taken from the excess monophosphate shunt in the production of G6PD dehydrogenase enzyme. So we need this enzyme in order to produce NADPH, and so there is protection of the cells. So in oxidative stress, if the patient has mutation in this enzyme, there would be damage to the cells, damage to the membranes of the cells, but also the oxygenated water is taking electrons from the hemoglobin, and so precipitated hemoglobin would look like Heinz bodies in the red blood cells. So the best initial test would be complete cell count, etc. But the patient is in the acute moment with a little bit of fatigue, back pain, and urine with red color. So there is not exactly treatment, just supportive therapy, but we need to avoid the stressors. Now, we need to avoid certainly the stressors and to confirm that the patient has glucose 6PD deficiency, dehydrogenase deficiency. This would be if we do a blood smear and in a special stain called violet crystal, we will see the Heinz bodies. But usually it takes like 1 to 2 weeks after the acute event to see them. So we will wait to 1 to 2 weeks. Now, the levels of glucose D6PD, glucose 6-phosphate dehydrogenase enzyme would be low, but in the acute event could be normal. So we need to wait 1 to 2 months because the half-life of a red blood cell is 3 months. Now, the half-life of this mutated glucose 6PD enzyme, it depends also if we're talking about the African or Mediterranean variant. The Mediterranean variant is more severe, so the half-life is less, and they present with more recurrent episodes of this. But we will measure this 1 to 2 months. So first, avoid the stressors, and next, check for glucose 6PD deficiency, but not at the moment of the stress, because the G6PD will be normal and sometimes will not see the Heinz bodies. So we need for the Heinz bodies to wait 1 to 2 weeks, and for the levels of the enzyme 1 to 2 months.